Molecular models show a DNA double helix that is packed in a chromosome in part A, and two proteins are shown in parts B and C. Figure 15. Genes, which are carried on chromosomes, are linearly organized instructions for making the RNA and protein molecules that are necessary for all of the processes of life. The B interleukin-2 protein and C alpha-2 globulin protein are just two examples of the array of different molecular structures that are encoded by genes. Credit chromosome. National Human Genome Research Institute. Credit interleukin-2. Raman Harati created from PDB-1M47 and rendered with Pymol. Credit alpha-2 globulin. Darren Logan rendered with AISMIG. Chapter outline. 15. 1. The genetic code. 15. 2. Prokaryotic transcription. 15. 3. Eukaryotic transcription. 15. 4. RNA processing in eukaryotes. 15. 5. Ribosomes and protein synthesis. Since the rediscovery of Mendel's work in 1900, the definition of the gene has progressed from an abstract unit of heredity to a tangible molecular entity capable of replication, expression, and mutation. Figure 15. 1. Genes are composed of DNA and are linearly arranged on chromosomes. Genes specify the sequences of amino acids, which are the building blocks of proteins. In turn, proteins are responsible for orchestrating nearly every function of the cell. Both genes and the proteins they encode are absolutely essential to life as we know it. Learning objectives. By the end of this section, you will be able to do the following. Explain the central dogma of DNA protein synthesis. Describe the genetic code and how the nucleotide sequence prescribes the amino acid and the protein sequence. The cellular process of transcription generates messenger RNA, mRNA, a mobile molecular copy of one or more genes with an alphabet of A, C, G, and uracil, U. Translation of the mRNA template on ribosomes converts nucleotide-based genetic information into a protein product. That is the central dogma of DNA protein synthesis. Protein sequences consist of 20 commonly occurring amino acids. Therefore, it can be said that the protein alphabet consists of 20 letters. Figure 15.2. Different amino acids have different chemistries, such as acidic versus basic, or polar and nonpolar, and different structural constraints. Variation in amino acid sequence is responsible for the enormous variation in protein structure and function. Structures of the 20 amino acids are given. 6 amino acids, glycine, alanine, valine, leucine, methionine, and isoleucine, have R groups that are nonpolar and aliphatic, meaning they do not have a ring. 6 amino acids, serine, threonine, cysteine, proline, asparagine, and glutamate, have R groups that are polar but uncharged. 3 amino acids, lysine, arginine, and histidine, have R groups that are positively charged. 2 amino acids, glutamate and aspartate, have R groups that are negatively charged. 3 amino acids, phenylalanine, tyrosine, and tryptophan, have nonpolar and aromatic, meaning they have a ring R groups. Figure 15. 2 structures of the 20 amino acids found in proteins are shown. Each amino acid is composed of an amino group, NH plus 3, a carboxyl group Q, and a side chain blue. The side chain may be nonpolar, polar, or charged, as well as large or small. It is the variety of amino acid side chains that gives rise to the incredible variation of protein structure and function. The central dogma. DNA encodes RNA. RNA encodes protein. The flow of genetic information in cells, from DNA to mRNA to protein, is described by the central dogma, figure 15. 3, which states that genes specify the sequence of mRNAs, which in turn specify the sequence of amino acids making up all proteins. The decoding of one molecule to another is performed by specific proteins and RNAs. Because the information stored in DNA is so central to cellular function, it makes intuitive sense that the cell would make mRNA copies of this information for protein synthesis, while keeping the DNA itself intact and protected. The copying of DNA to RNA is relatively straightforward, with one nucleotide being added to the mRNA strand for every nucleotide read in the DNA strand. The translation to protein is a bit more complex because 3 mRNA nucleotides correspond to one amino acid in the polypeptide sequence. However, the translation to protein is still systematic and collinear, such that nucleotides 1 to 3 correspond to amino acid 1, nucleotides 4 to 6 correspond to amino acid 2, and so on. To make a protein, genetic information encoded by the DNA must be transcribed onto an mRNA molecule. The RNA is then processed by splicing to remove exons and by the addition of a 5' cap and a poly A tail. A ribosome then reads the sequence on the mRNA and uses this information to string amino acids into a protein. Figure 15. 3 instructions on DNA are transcribed onto messenger RNA. Ribosomes are able to read the genetic information inscribed on a strand of messenger RNA and use this information to string amino acids together into a protein. The genetic code is degenerate and universal. Each amino acid is defined by a 3-nucleotide sequence called the triplet codon. Given the different numbers of letters in the mRNA and protein alphabets, scientists theorized that single amino acids must be represented by combinations of nucleotides. Nucleotide doublets would not be sufficient to specify every amino acid because there are only 16 possible 2-nucleotide combinations, 42. In contrast, there are 64 possible nucleotide triplets, 43, which is far more than the number of amino acids. Scientists theorized that amino acids were encoded by nucleotide triplets and that the genetic code was degenerate. In other words, a given amino acid could be encoded by more than one nucleotide triplet. This was later confirmed experimentally. Francis Crick and Sidney Brenner used the chemical mutagen proflavin to insert 1, 2, or 3 nucleotides into the gene of a virus. When 1 or 2 nucleotides were inserted, the normal proteins were not produced. When 3 nucleotides were inserted, the protein was synthesized and functional. This demonstrated that the amino acids must be specified by groups of 3 nucleotides. These nucleotide triplets are called codons. The insertion of 1 or 2 nucleotides completely changed the triplet reading frame, thereby altering the message for every subsequent amino acid, figure 15. 5. Though insertion of 3 nucleotides caused an extra amino acid to be inserted during translation, the integrity of the rest of the protein was maintained. Scientists painstakingly solved the genetic code by translating synthetic mRNAs in vitro and sequencing the proteins they specified, figure 15. 4. Figure shows all 64 codons. 61 of these code for amino acids, and 3 are stop codons. Figure 15. For this figure shows the genetic code for translating each nucleotide triplet in mRNA into an amino acid or a termination signal in a protein. Credit, modification of work by NIH. In addition to codons that instruct the addition of a specific amino acid to a polypeptide chain, 3 of the 64 codons terminate protein synthesis and release the polypeptide from the translation machinery. These triplets are called nonsense codons or stop codons. Another codon, AUG, also has a special function. In addition to specifying the amino acid methionine, it also serves as the start codon to initiate translation. The reading frame for translation is set by the AUG start codon near the 5' end of the mRNA. Following the start codon, the mRNA is read in groups of 3 until a stop codon is encountered. The arrangement of the coding table reveals the structure of the code. There are 16 blocks of codons, each specified by the first and second nucleotides of the codons within the block, e.g., the AC block that corresponds to the amino acid threonine THR. Some blocks are divided into a pyrimidine half, in which the codon ends with U or C, and a purine half, in which the codon ends with A or G. Some amino acids get a whole block of 4 codons, like alanine, ALA, threonine, THR, and proline, PRO. Some get the pyrimidine half of their block, like histidine, HIS, and asparagine, ASIN. Others get the purine half of their block, like glutamate, GLU, and lysine, LYS. Note that some amino acids get a block and a half block for a total of 6 codons. The specification of a single amino acid by multiple similar codons is called degeneracy. Degeneracy is believed to be a cellular mechanism to reduce the negative impact of random mutations. Codons that specify the same amino acid typically only differ by one nucleotide. In addition, amino acids with chemically similar side chains are encoded by similar codons. For example, aspartate asp and glutamate glu, which occupy the Ga* block, are both negatively charged. This nuance of the genetic code ensures that a single nucleotide substitution mutation might specify the same amino acid but have no effect or specify a similar amino acid, preventing the protein from being rendered completely non-functional. The genetic code is nearly universal. With a few minor exceptions, virtually all species use the same genetic code for protein synthesis. Conservation of codons means that a purified mRNA encoding the globin protein in horses could be transferred to a tulip cell, and the tulip would synthesize horse globin. That there is only one genetic code is powerful evidence that all of life on Earth shares a common origin, especially considering that there are about 1084 possible combinations of 20 amino acids and 64 triplet codons. Link to learning. Transcribe a gene and translate it to protein using complementary pairing and the genetic code at this site. Illustration shows a frameshift mutation in which the reading frame is altered by the deletion of 2 amino acids. Figure 15. 5. The deletion of 2 nucleotides shifts the reading frame of an mRNA and changes the entire protein message, creating a non-functional protein or terminating protein synthesis altogether. Scientific method connection, which has more DNA, a kiwi or a strawberry. Photographs show a thin slice of a green kiwi fruit and a bowl of strawberries. Figure 15. 6. Do you think that a kiwi or a strawberry has more DNA per fruit? Credit kiwi, Kelby/Flicker. Credit strawberry, Alistair McDiarmid. Question: would a kiwi and strawberry that are approximately the same size, figure 15. 6, also have approximately the same amount of DNA? Background: genes are carried on chromosomes and are made of DNA. All mammals are diploid, meaning they have 2 copies of each chromosome. However, not all plants are diploid. The common strawberry is octoploid, 8N, and the cultivated kiwi is hexaploid, 6N. Research the total number of chromosomes in the cells of each of these fruits, and think about how this might correspond to the amount of DNA in these fruits, cell nuclei. What other factors might contribute to the total amount of DNA in a single fruit? Read about the technique of DNA isolation to understand how each step in the isolation protocol helps liberate and precipitate DNA. Hypothesis: hypothesize whether you would be able to detect a difference in DNA quantity from similarly sized strawberries and kiwis. Which fruit do you think would yield more DNA? Test your hypothesis. Isolate the DNA from a strawberry and a kiwi that are similarly sized. Perform the experiment in at least triplicate for each fruit. Prepare a bottle of DNA extraction buffer from 900 ml water, 50 ml dish detergent, and 2 teaspoons of table salt. Mix by inversion, cap it and turn it upside down a few times. Grind a strawberry and a kiwi by hand in a plastic bag, or using a mortar and pestle, or with a metal bowl and the end of a blunt instrument. Grind for at least 2 minutes per fruit. Add 10 ml of the DNA extraction buffer to each fruit and mix well for at least 1 minute. Remove cellular debris by filtering each fruit mixture through cheesecloth or porous cloth and into a funnel placed in a test tube or an appropriate container. Pour ice-cold ethanol or isopropanol rubbing alcohol into the test tube. You should observe white, precipitated DNA. Gather the DNA from each fruit by winding it around separate glass rods. Record your observations. Because you are not quantitatively measuring DNA volume, you can record for each trial whether the 2 fruits produce the same or different amounts of DNA as observed by eye. If one or the other fruit produced noticeably more DNA, record this as well. Determine whether your observations are consistent with several pieces of each fruit. Analyze your data. Did you notice an obvious difference in the amount of DNA produced by each fruit? Were your results reproducible? Draw a conclusion. Given what you know about the number of chromosomes in each fruit, can you conclude that chromosome number necessarily correlates to DNA amount? Can you identify any drawbacks to this procedure? If you had access to a laboratory, how could you standardize your comparison and make it more quantitative? Learning objectives. By the end of this section, you will be able to do the following. List the different steps in prokaryotic transcription. Discuss the role of promoters in prokaryotic transcription. Describe how and when transcription is terminated. The prokaryotes, which include bacteria and archaea, are mostly single-celled organisms that, by definition, lack membrane-bound nuclei and other organelles. A bacterial chromosome is a closed circle that, unlike eukaryotic chromosomes, is not organized around histone proteins. The central region of the cell in which prokaryotic DNA resides is called the nucleoid region. In addition, prokaryotes often have abundant plasmids, which are shorter circular DNA molecules that may only contain one or a few genes. Plasmids can be transferred independently of the bacterial chromosome during cell division and often carry traits such as those involved with antibiotic resistance. Transcription in prokaryotes and in eukaryotes requires the DNA double helix to partially unwind in the region of mRNA synthesis. The region of unwinding is called a transcription bubble. Transcription always proceeds from the same DNA strand for each gene, which is called the template strand. The mRNA product is complementary to the template strand and is almost identical to the other DNA strand, called the non-template strand or the coding strand. The only nucleotide difference is that in mRNA, all of the T nucleotides are replaced with U nucleotides, figure 15.7. In an RNA double helix, a can bind U via 2 hydrogen bonds, just as in A-T pairing in a DNA double helix. Illustration shows RNA synthesis by RNA polymerase. The RNA strand is synthesized in the 5' to 3' direction. Figure 15. 7 messenger RNA is a copy of protein coding information in the coding strand of DNA, with the substitution of U in the RNA for T in the coding sequence. However, new RNA nucleotides base pair with the nucleotides of the template strand. RNA is synthesized in its 5' direction using the enzyme RNA polymerase. As the template is read, the DNA unwinds ahead of the polymerase and then rewinds behind it. The nucleotide pair in the DNA double helix that corresponds to the site from which the first 5 mRNA nucleotide is transcribed is called the +1 site or the initiation site. Nucleotides preceding the initiation site are denoted with a and are designated upstream nucleotides. Conversely, nucleotides following the initiation site are denoted with plus numbering and are called downstream nucleotides. Initiation of transcription in prokaryotes. Prokaryotes do not have membrane-enclosed nuclei. Therefore, the processes of transcription, translation, and mRNA degradation can all occur simultaneously. The intracellular level of a bacterial protein can quickly be amplified by multiple transcription and translation events that occur concurrently on the same DNA template. Prokaryotic genomes are very compact, and prokaryotic transcripts often cover more than one gene or cistron, a coding sequence for a single protein. Polycistronic mRNAs are then translated to produce more than one kind of protein. Our discussion here will exemplify transcription by describing this process in Escherichia coli, a well-studied eubacterial species. Although some differences exist between transcription in E. coli and transcription in archaea, an understanding of E. coli transcription can be applied to virtually all bacterial species. Prokaryotic RNA polymerase. Prokaryotes use the same RNA polymerase to transcribe all of their genes. In E. coli, the polymerase is composed of 5 polypeptide subunits, 2 of which are identical. 4 of these subunits, denoted alpha, alpha, beta, and beta, comprise the polymerase core enzyme. These subunits assemble every time a gene is transcribed, and they disassemble once transcription is complete. Each subunit has a unique role. The 2 alpha subunits are necessary to assemble the polymerase on the DNA. The beta subunit binds to the ribonucleoside triphosphate that will become part of the nascent mRNA molecule. And the beta subunit binds the DNA template strand. The 5th subunit, sigma, is involved only in transcription initiation. It confers transcriptional specificity such that the polymerase begins to synthesize mRNA from an appropriate initiation site. Without sigma, the core enzyme would transcribe from random sites and would produce mRNA molecules that specified protein gibberish. The polymerase comprised of all 5 subunits is called the holoenzyme. Prokaryotic promoters. A promoter is a DNA sequence onto which the transcription machinery, including RNA polymerase, binds and initiates transcription. In most cases, promoters exist upstream of the genes they regulate. The specific sequence of a promoter is very important because it determines whether the corresponding gene is transcribed all the time, some of the time, or infrequently. Although promoters vary among prokaryotic genomes, a few elements are evolutionarily conserved in many species. At the -10 and -35 regions upstream of the initiation site, there are 2 promoter consensus sequences, or regions that are similar across all promoters and across various bacterial species. Figure 15. 8. The -10 sequence, called the -10 region, has the consensus sequence TATAAT. The -35 sequence has the consensus sequence TTGACA. These consensus sequences are recognized and bound by sigma. Once this interaction is made, the subunits of the core enzyme bind to the site. The T-rich -10 region facilitates unwinding of the DNA template, and several phosphodiester bonds are made. The transcription initiation phase ends with the production of abortive transcripts, which are polymers of approximately 10 nucleotides that are made and released. Illustration shows the sigma subunit of Rn, a polymerase bound to 2 consensus sequences that are 10 and 35 bases upstream of the transcription start site. Rn, a polymerase, is bound to sigma. Figure 15. 8. The sigma subunit of prokaryotic RNA polymerase recognizes consensus sequences found in the promoter region upstream of the transcription start site. The sigma subunit dissociates from the polymerase after transcription has been initiated. Link to learning. View this molecular movie's animation to see the transcription process as it happens in the cell. Elongation and termination in prokaryotes. The transcription elongation phase begins with the release of the sigma subunit from the polymerase. The dissociation of sigma allows the core enzyme to proceed along the DNA template, synthesizing mRNA in the 5 to 3 direction at a rate of approximately 40 nucleotides per second. As elongation proceeds, the DNA is continuously unwound ahead of the core enzyme and rewound behind it. The base pairing between DNA and RNA is not stable enough to maintain the stability of the mRNA synthesis components. Instead, the RNA polymerase acts as a stable linker between the DNA template and the nascent RNA strands to ensure that elongation is not interrupted prematurely. Prokaryotic termination signals. Once a gene is transcribed, the prokaryotic polymerase needs to be instructed to dissociate from the DNA template and liberate the newly made mRNA. Depending on the gene being transcribed, there are 2 kinds of termination signals. One is protein-based and the other is RNA-based. Rho-dependent termination is controlled by the Rho protein, which tracks along behind the polymerase on the growing mRNA chain. Near the end of the gene, the polymerase encounters a run of G nucleotides on the DNA template and it stalls. As a result, the Rho protein collides with the polymerase. The interaction with Rho releases the mRNA from the transcription bubble. Rho-independent termination is controlled by specific sequences in the DNA template strand. As the polymerase nears the end of the gene being transcribed, it encounters a region rich in G nucleotides. The mRNA folds back on itself, and the complementary G nucleotides bind together. The result is a stable hairpin that causes the polymerase to stall as soon as it begins to transcribe a region rich in T nucleotides. The complementary U, a region of the mRNA transcript, forms only a weak interaction with the template DNA. This, coupled with the stalled polymerase, induces enough instability for the core enzyme to break away and liberate the new mRNA transcript. Upon termination, the process of transcription is complete. By the time termination occurs, the prokaryotic transcript would already have been used to begin synthesis of numerous copies of the encoded protein, because these processes can occur concurrently. The unification of transcription, translation, and even mRNA degradation is possible because all of these processes occur in the same 5 to 3 direction. And because there is no membranous compartmentalization in the prokaryotic cell, figure 15. 9. In contrast, the presence of a nucleus in eukaryotic cells precludes simultaneous transcription and translation. Illustration shows a portion of a DNA molecule. Multiple strands of mRNA hang off of the DNA at the locations where mRNA is being transcribed. Each mRNA strand has multiple ribosomes attached at different locations along its length, creating proteins. One mRNA strand with multiple ribosomes attached to it is labeled a polyribosome. Figure 15. 9 multiple polymerases can transcribe a single bacterial gene, while numerous ribosomes concurrently translate the mRNA transcripts into polypeptides. In this way, a specific protein can rapidly reach a high concentration in the bacterial cell. Link to learning. Visit this BioStudio animation to see the process of prokaryotic transcription. Learning objectives. By the end of this section, you will be able to do the following. List the steps in eukaryotic transcription. Discuss the role of RNA polymerases in transcription. Compare and contrast the 3 RNA polymerases. Explain the significance of transcription factors. Prokaryotes and eukaryotes perform fundamentally the same process of transcription, with a few key differences. The most important difference between prokaryote and eukaryote transcription is due to the latter's membrane-bound nucleus and organelles. With the genes bound in a nucleus, the eukaryotic cell must be able to transport its mRNA to the cytoplasm and must protect its mRNA from degrading before it is translated. Eukaryotes also employ 3 different polymerases that each transcribe a different subset of genes. Eukaryotic mRNAs are usually monogenic, meaning that they specify a single protein. Initiation of transcription in eukaryotes. Unlike the prokaryotic polymerase that can bind to a DNA template on its own, eukaryotes require several other proteins, called transcription factors, to first bind to the promoter region and then to help recruit the appropriate polymerase. The 3 eukaryotic RNA polymerases. The features of eukaryotic mRNA synthesis are markedly more complex than those of prokaryotes. Instead of a single polymerase comprising 5 subunits, the eukaryotes have 3 polymerases that are each made up of 10 subunits or more. Each eukaryotic polymerase also requires a distinct set of transcription factors to bring it to the DNA template. RNA polymerase I is located in the nucleolus, a specialized nuclear substructure in which ribosomal RNA, rRNA, is transcribed, processed, and assembled into ribosomes, Table 15. 1. The rRNA molecules are considered structural RNAs because they have a cellular role but are not translated into protein. The rRNAs are components of the ribosome and are essential to the process of translation. RNA polymerase I synthesizes all of the rRNAs from the tandemly duplicated set of 18S, 5, 8S, and 28S ribosomal genes. Note that the S designation applies to Svedberg units, a non-additive value that characterizes the speed at which a particle sediments during centrifugation. Locations, products, and sensitivities of the 3 eukaryotic RNA polymerases. RNA polymerase cellular compartment product of transcription alpha-amanitin sensitivity. Nucleolus all rRNAs, except 5 srRNA insensitive. Roman 2 nucleus all protein coding nuclear pre-mRNAs extremely sensitive. Roman 3 nucleus 5s rRNA, tRNAs, and small nuclear RNAs moderately sensitive. Table 15. 1. RNA polymerase II is located in the nucleus and synthesizes all protein-coding nuclear pre-mRNAs. Eukaryotic pre-mRNAs undergo extensive processing after transcription, but before translation. For clarity, this module's discussion of transcription and translation in eukaryotes will use the term mRNAs to describe only the mature, processed molecules that are ready to be translated. RNA polymerase II is responsible for transcribing the overwhelming majority of eukaryotic genes. RNA polymerase III is also located in the nucleus. This polymerase transcribes a variety of structural RNAs that includes the 5S pre-rRNA, transfer pre-RNAs, pre-tRNAs, and small nuclear pre-RNAs. The tRNAs have a critical role in translation. They serve as the adapter molecules between the mRNA template and the growing polypeptide chain. Small nuclear RNAs have a variety of functions, including splicing, pre-mRNAs, and regulating transcription factors. A scientist characterizing a new gene can determine which polymerase transcribes it by testing whether the gene is expressed in the presence of alpha-amanitin, an oligopeptide toxin produced by the fly agaric toadstool mushroom and other species of Amanita. Interestingly, the alpha-amanitin affects the three polymerases very differently. Table 15.1. RNA polymerase I is completely insensitive to alpha-amanitin, meaning that the polymerase can transcribe DNA in vitro in the presence of this poison. RNA polymerase III is moderately sensitive to the toxin. In contrast, RNA polymerase II is extremely sensitive to alpha-amanitin. The toxin prevents the enzyme from progressing down the DNA and thus inhibits transcription. Knowing the transcribing polymerase can provide clues as to the general function of the gene being studied. Because RNA polymerase II transcribes the vast majority of genes, we will focus on this polymerase in our subsequent discussions about eukaryotic transcription factors and promoters. RNA polymerase II promoters and transcription factors. Eukaryotic promoters are much larger and more intricate than prokaryotic promoters. However, both have a sequence similar to the -10 sequence of prokaryotes. In eukaryotes, this sequence is called the TATA box and has the consensus sequence TATAA on the coding strand. It is located at -25 to -35 bases relative to the initiation, +1, cite figure 15.10. This sequence is not identical to the E. coli -10 box, but it conserves the A-T-rich element. The thermostability of A-T bonds is low, and this helps the DNA template to locally unwind in preparation for transcription. Instead of the simple sigma factor that helps bind the prokaryotic RNA polymerase to its promoter, eukaryotes assemble a complex of transcription factors required to recruit RNA polymerase II to a protein-coding gene. Transcription factors that bind to the promoter are called basal transcription factors. These basal factors are all called TFII for transcription factor polymerase II plus an additional letter, AJ. The core complex is TFIID, which includes a TATA binding protein, TBP. The other transcription factors systematically fall into place on the DNA template, with each one further stabilizing the pre-initiation complex and contributing to the recruitment of RNA polymerase II. Visual connection. Illustration shows a series of transcription factors binding to the promoter, which is upstream of the gene. After all of the transcription factors are bound, RNA polymerase II binds as well. Figure 15.10. A generalized promoter of a gene transcribed by RNA polymerase II is shown. Transcription factors recognize the promoter. RNA polymerase II then binds and forms the transcription initiation complex. A scientist splices a eukaryotic promoter in front of a bacterial gene and inserts the gene in a bacterial chromosome. Would you expect the bacteria to transcribe the gene? Some eukaryotic promoters also have a conserved CAAT box, GGCATCT, at approximately. Further upstream of the TATA box, eukaryotic promoters may also contain one or more GC-rich boxes, GGCG, or octamer boxes, ATTTGCAT. These elements bind cellular factors that increase the efficiency of transcription initiation and are often identified in more active genes that are constantly being expressed by the cell. Basal transcription factors are crucial in the formation of a pre-initiation complex on the DNA template that subsequently recruits RNA polymerase II for transcription initiation. The complexity of eukaryotic transcription does not end with the polymerases and promoters. An army of other transcription factors, which bind to upstream enhancers and silencers, also help to regulate the frequency with which pre-mRNA is synthesized from a gene. Enhancers and silencers affect the efficiency of transcription, but are not necessary for transcription to proceed. Promoter structures for RNA polymerases I and III. The processes of bringing RNA polymerases I and III to the DNA template involve slightly less complex collections of transcription factors, but the general theme is the same. The conserved promoter elements for genes transcribed by polymerases I and III differ from those transcribed by RNA polymerase II. RNA polymerase I transcribes genes that have two GC-rich promoter sequences in the -45 to +20 region. These sequences alone are sufficient for transcription initiation to occur. But promoters with additional sequences in the region from -180 to -105 upstream of the initiation site will further enhance initiation. Genes that are transcribed by RNA polymerase III have upstream promoters or promoters that occur within the genes themselves. Eukaryotic transcription is a tightly regulated process that requires a variety of proteins to interact with each other and with the DNA strand. Although the process of transcription in eukaryotes involves a greater metabolic investment than in prokaryotes, it ensures that the cell transcribes precisely the pre-mRNAs that it needs for protein synthesis. Evolution connection. The evolution of promoters. The evolution of genes may be a familiar concept. Mutations can occur in genes during DNA replication, and the result may or may not be beneficial to the cell. By altering an enzyme, structural protein, or some other factor, the process of mutation can transform functions or physical features. However, eukaryotic promoters and other gene regulatory sequences may evolve as well. For instance, consider a gene that, over many generations, becomes more valuable to the cell. Maybe the gene encodes a structural protein that the cell needs to synthesize in abundance for a certain function. If this is the case, it would be beneficial to the cell for that gene's promoter to recruit transcription factors more efficiently and increase gene expression. Scientists examining the evolution of promoter sequences have reported varying results. In part, this is because it is difficult to infer exactly where a eukaryotic promoter begins and ends. Some promoters occur within genes, others are located very far upstream or even downstream of the genes they are regulating. However, when researchers limited their examination to human core promoter sequences that were defined experimentally as sequences that bind the pre-initiation complex, they found that promoters evolve even faster than protein-coding genes. It is still unclear how promoter evolution might correspond to the evolution of humans or other complex organisms. However, the evolution of a promoter to effectively make more or less of a given gene product is an intriguing alternative to the evolution of the genes themselves. 1. Eukaryotic elongation and termination. Following the formation of the pre-initiation complex, the polymerase is released from the other transcription factors, and elongation is allowed to proceed as it does in prokaryotes with the polymerase synthesizing pre-mRNA in the 5 to 3 direction. As discussed previously, RNA polymerase II transcribes the major share of eukaryotic genes. So in this section, we will focus on how this polymerase accomplishes elongation and termination. Although the enzymatic process of elongation is essentially the same in eukaryotes and prokaryotes, the DNA template is considerably more complex. When eukaryotic cells are not dividing, their genes exist as a diffuse mass of DNA and proteins called chromatin. The DNA is tightly packaged around charged histone proteins at repeated intervals. These DNA, histone complexes, collectively called nucleosomes, are regularly spaced and include 146 nucleotides of DNA wound around 8 histones like thread around a spool. For polynucleotide synthesis to occur, the transcription machinery needs to move histones out of the way every time it encounters a nucleosome. This is accomplished by a special protein complex called FACT, which stands for Facilitates Chromatin Transcription. This complex pulls histones away from the DNA template as the polymerase moves along it. Once the pre-mRNA is synthesized, the FACT complex replaces the histones to recreate the nucleosomes. The termination of transcription is different for the different polymerases. Unlike in prokaryotes, elongation by RNA polymerase II in eukaryotes takes place 1,000 to 2,000 nucleotides beyond the end of the gene being transcribed. This pre-mRNA tail is subsequently removed by cleavage during mRNA processing. On the other hand, RNA polymerases I and III require termination signals. Genes transcribed by RNA polymerase I contain a specific 18 nucleotide sequence that is recognized by a termination protein. The process of termination in RNA polymerase III involves an mRNA hairpin similar to row-independent termination of transcription in prokaryotes. Learning objectives. By the end of this section, you will be able to do the following. Describe the different steps in RNA processing. Understand the significance of exons, introns, and splicing for mRNAs. Explain how tRNAs and rRNAs are processed. After transcription, eukaryotic pre-mRNAs must undergo several processing steps before they can be translated. Eukaryotic and prokaryotic. tRNAs and rRNAs also undergo processing before they can function as components in the protein synthesis machinery. mRNA processing. The eukaryotic pre-mRNA undergoes extensive processing before it is ready to be translated. Eukaryotic protein coding sequences are not continuous, as they are in prokaryotes. The coding sequences, exons, are interrupted by non-coding introns, which must be removed to make a translatable mRNA. The additional steps involved in eukaryotic mRNA maturation also create a molecule with a much longer half-life than a prokaryotic mRNA. Eukaryotic mRNAs last for several hours, whereas the typical E. coli mRNA lasts no more than 5 seconds. Pre-mRNAs are first coded in RNA-stabilizing proteins. These protect the pre-mRNA from degradation while it is processed and exported out of the nucleus. The 3 most important steps of pre-mRNA processing are the addition of stabilizing and signaling factors at the 5 and 3 ends of the molecule and the removal of the introns, figure 15. 11. In rare cases, the mRNA transcript can be edited after it is transcribed. An illustration shows that in pre-mRNA processing, there is a primary RNA transcript including regions labeled, left to right, as exon 1, intron, exon 2, intron, and exon 3. The introns are cut and exons are spliced together to produce mRNA. After RNA processing, there is a spliced RNA with these parts. Left to right are a 5' cap, a 5' untranslated region, exon 1, exon 2, exon 3, a 3' untranslated region, and a poly a tail. The 5' untranslated region is not separate from exon 1, but is the beginning of exon 1. The 3' untranslated region is not separate from exon 3, but the final part of exon 3. Figure 15. 11 Eukaryotic pre-mRNA processing. In addition to 5' cap and 3' poly a tail addition, introns must be precisely removed and exons joined to generate a functional mRNA. Nucleotides upstream, towards the 5' cap of the translation start codon, are part of the 5' untranslated region 5 UTR. Nucleotides downstream, towards 3' end of the stop codon form the 3 UTR. Both 5 and 3 UTRs are important for regulating translation initiation and mRNA stability. Credit: Rao, A. Ryan, K. Fletcher, S. and Tag, A. Department of Biology, Texas A&M University. Evolution connection. RNA editing in trypanosomes. The trypanosomes are a group of protozoa that include the pathogen Trypanosoma brucei, which causes nagana in cattle and sleeping sickness in humans throughout great areas of Africa. Figure 15. 12. The trypanosome is carried by biting flies in the genus Glossina, commonly called tsetse flies. Trypanosomes, and virtually all other eukaryotes, have organelles called mitochondria that supply the cell with chemical energy. Mitochondria are organelles that express their own DNA and are believed to be the remnants of a symbiotic relationship between a eukaryote and an engulfed prokaryote. The mitochondrial DNA of trypanosomes exhibit an interesting exception to the central dogma. Their pre-mRNAs do not have the correct information to specify a functional protein. Usually, this is because the mRNA is missing several eunucleotides. The cell performs an additional RNA processing step called RNA editing to remedy this. Micrograph shows T. brucei, which has a U-shaped cell body and a long tail. Figure 15. 12 Trypanosoma brucei is the causative agent of sleeping sickness in humans. The mRNAs of this pathogen must be modified by the addition of nucleotides before protein synthesis can occur. Credit: modification of work by Torsten Otzenrader. Other genes in the mitochondrial genome encode 40 to 80 nucleotide guide RNAs. One or more of these molecules interacts by complementary base pairing with some of the nucleotides in the pre-mRNA transcript. However, the guide RNA has more nucleotides than the pre-mRNA has U-nucleotides with which to bind. In these regions, the guide RNA loops out. The three ends of guide RNAs have a long poly-U tail, and these U bases are inserted in regions of the pre-mRNA transcript at which the guide RNAs are looped. This process is entirely mediated by RNA molecules. That is, guide RNAs, rather than proteins, serve as the catalysts in RNA editing. RNA editing is not just a phenomenon of trypanosomes. In the mitochondria of some plants, almost all pre-mRNAs are edited. RNA editing has also been identified in mammals such as rats, rabbits, and even humans. What could be the evolutionary reason for this additional step in pre-mRNA processing? One possibility is that the mitochondria, being remnants of ancient prokaryotes, have an equally ancient RNA-based method for regulating gene expression. In support of this hypothesis, edits made to pre-mRNAs differ depending on cellular conditions. Although speculative, the process of RNA editing may be a holdover from a primordial time when RNA molecules, instead of proteins, were responsible for catalyzing reactions. 5. Capping. While the pre-mRNA is still being synthesized, a 7-methylguanosine cap is added to the 5' end of the growing transcript by a phosphate linkage. This functional group protects the nascent mRNA from degradation. In addition, factors involved in protein synthesis recognize the cap to help initiate translation by ribosomes. 3. Poly-A tail. Once elongation is complete, the pre-mRNA is cleaved by an endonuclease between an AAUAA consensus sequence and a G-URH sequence, leaving the AAUAA sequence on the pre-mRNA. An enzyme called poly-A polymerase then adds a string of approximately 200 residues, called the poly-A tail. This modification further protects the pre-mRNA from degradation and is also the binding site for a protein necessary for exporting the processed mRNA to the cytoplasm. Pre-mRNA splicing. Eukaryotic genes are composed of exons, which correspond to protein coding sequences. Exon signifies that they are expressed, and intervening sequences called introns. Intron denotes their intervening role, which may be involved in gene regulation but are removed from the pre-mRNA during processing. Intron sequences in mRNA do not encode functional proteins. The discovery of introns came as a surprise to researchers in the 1970s who expected that pre-mRNAs would specify protein sequences without further processing, as they had observed in prokaryotes. The genes of higher eukaryotes very often contain one or more introns. These regions may correspond to regulatory sequences. However, the biological significance of having many introns or having very long introns in a gene is unclear. It is possible that introns slow down gene expression because it takes longer to transcribe pre-mRNAs with lots of introns. Alternatively, introns may be non-functional sequence remnants left over from the fusion of ancient genes throughout the course of evolution. This is supported by the fact that separate exons often encode separate protein subunits or domains. For the most part, the sequences of introns can be mutated without ultimately affecting the protein product. All of a pre-mRNA's introns must be completely and precisely removed before protein synthesis. If the process errs by even a single nucleotide, the reading frame of the rejoined exons would shift, and the resulting protein would be dysfunctional. The process of removing introns and reconnecting exons is called splicing, figure 1513. Introns are removed and degraded while the pre-mRNA is still in the nucleus. Splicing occurs by a sequence-specific mechanism that ensures introns will be removed and exons rejoined with the accuracy and precision of a single nucleotide. Although the intron itself is non-coding, the beginning and end of each intron is marked with specific nucleotides, GU at the 5' end and AG at the 3' end of the intron. The splicing of pre-mRNAs is conducted by complexes of proteins and RNA molecules called spliceosomes. Visual connection. Illustration shows a spliceosome bound to mRNA. An intron is wrapped around small rn as associated with the spliceosome. When the splice is complete, The exons on either side of the intron are fused together, and the intron forms a ring structure. Figure 15. 13 pre-mRNA splicing involves the precise removal of introns from the primary RNA transcript. The splicing process is catalyzed by protein complexes called spliceosomes that are composed of proteins and RNA molecules called small nuclear RNAs, snRNA. Spliceosomes recognize sequences at the 5 and 3 end of the intron. Rao, A. and Ryan, K. Department of Biology, Texas A&M University. Errors in splicing are implicated in cancers and other human diseases. What kinds of mutations might lead to splicing errors? Think of different possible outcomes if splicing errors occur. Note that more than 70 individual introns can be present, and each has to undergo the process of splicing, in addition to 5, capping, and the addition of a poly-A tail, just to generate a single, translatable mRNA molecule. Link to learning. See how introns are removed during RNA splicing at this website. Processing of tRNAs and rRNAs. The tRNAs and rRNAs are structural molecules that have roles in protein synthesis. However, these RNAs are not themselves translated. Pre-rRNAs are transcribed, processed, and assembled into ribosomes in the nucleolus. Pre-tRNAs are transcribed and processed in the nucleus and then released into the cytoplasm where they are linked to free amino acids for protein synthesis. Most of the tRNAs and rRNAs in eukaryotes and prokaryotes are first transcribed as a long precursor molecule that spans multiple rRNAs or tRNAs. Enzymes then cleave the precursors into subunits corresponding to each structural RNA. Some of the bases of pre-rRNAs are methylated. That is, a CH3 methyl functional group is added for stability. Pre-tRNA molecules also undergo methylation. As with pre-mRNAs, subunit excision occurs in eukaryotic pre-RNAs destined to become tRNAs or rRNAs. Mature rRNAs make up approximately 50% of each ribosome. Some of a ribosome's RNA molecules are purely structural, whereas others have catalytic or binding activities. Mature tRNAs take on a three-dimensional structure through local regions of base pairing stabilized by intramolecular hydrogen bonding. The tRNA folds to position the amino acid binding site at one end and the anticodon at the other end, figure 15. 14. The anticodon is a 3-nucleotide sequence in a tRNA that interacts with an mRNA codon through complementary base pairing. The molecular model of phenylalanine tRNA is L-shaped. At one end is the anticodon, A-G at the other end is the attachment site for the amino acid phenylalanine. Figure 15. 14. This is a space-filling model of a tRNA molecule that adds the amino acid phenylalanine to a growing polypeptide chain. The anticodon AAG binds the codon UUC on the mRNA. The amino acid phenylalanine is attached to the other end of the tRNA. Learning objectives. By the end of this section, you will be able to do the following. Describe the different steps in protein synthesis. Discuss the role of ribosomes in protein synthesis. The synthesis of proteins consumes more of a cell's energy than any other metabolic process. In turn, proteins account for more mass than any other component of living organisms, with the exception of water, and proteins perform virtually every function of a cell. The process of translation, or protein synthesis, involves the decoding of an mRNA message into a polypeptide product. Amino acids are covalently strung together by interlinking peptide bonds in lengths ranging from approximately 50 to more than 1,000 amino acid residues. Each individual amino acid has an amino group, NH2, and a carboxyl, COOH group. Polypeptides are formed when the amino group of one amino acid forms an amide, i.e., peptide bond with the carboxyl group of another amino acid, figure 1515. This reaction is catalyzed by ribosomes and generates one water molecule. Illustration shows two amino acids side by side. Each amino acid has an amino group, a carboxyl group, and a side chain labeled R or R'. Upon formation of a peptide bond, the amino group is joined to the carboxyl group. A water molecule is released in the process. Figure 15. 15. a peptide bond links the carboxyl end of one amino acid with the amino end of another, producing one water molecule during the process. For simplicity in this image, only the functional groups involved in the peptide bond are shown. The R and R designations refer to the rest of each amino acid structure. The protein synthesis machinery. In addition to the mRNA template, many molecules and macromolecules contribute to the process of translation. The composition of each component may vary across species. For example, ribosomes may consist of different numbers of rRNAs and polypeptides depending on the organism. However, the general structures and functions of the protein synthesis machinery are comparable from bacteria to human cells. Translation requires the input of an mRNA template, ribosomes, tRNAs, and various enzymatic factors. Note, a ribosome can be thought of as an enzyme whose amino acid binding sites are specified by mRNA. Link to learning. Click through the steps of this PBS Interactive to see protein synthesis in action. Ribosomes. Even before an mRNA is translated, a cell must invest energy to build each of its ribosomes. In E. coli, there are between 10,000 and 70,000 ribosomes present in each cell at any given time. A ribosome is a complex macromolecule composed of structural and catalytic rRNAs and many distinct polypeptides. In eukaryotes, the nucleolus is completely specialized for the synthesis and assembly of rRNAs. Ribosomes exist in the cytoplasm of prokaryotes and in the cytoplasm and rough endoplasmic reticulum of eukaryotes. Mitochondria and chloroplasts also have their own ribosomes in the matrix and stroma, which look more similar to prokaryotic ribosomes and have similar drug sensitivities than the ribosomes just outside their outer membranes in the cytoplasm. Ribosomes dissociate into large and small subunits when they are not synthesizing proteins and reassociate during the initiation of translation. In E. coli, the small subunit is described as 30S, and the large subunit is 50S, for a total of 70S. Recall that Svedberg units are not additive. Mammalian ribosomes have a small 40S subunit and a large 60S subunit. For a total of 80S, the small subunit is responsible for binding the mRNA template, whereas the large subunit sequentially binds tRNAs. Each mRNA molecule is simultaneously translated by many ribosomes, all synthesizing protein in the same direction, reading the mRNA from 5 to 3 and synthesizing the polypeptide from the N-terminus to the C-terminus. The complete mRNA polyribosome structure is called a polysome. tRNAs. The tRNAs are structural RNA molecules that were transcribed from genes by RNA polymerase III. Depending on the species, 40 to 60 types of tRNAs exist in the cytoplasm. Transfer RNAs serve as adapter molecules. Each tRNA carries a specific amino acid and recognizes one or more of the mRNA codons that define the order of amino acids in a protein. Aminosyl tRNAs bind to the ribosome and add the corresponding amino acid to the polypeptide chain. Therefore, tRNAs are the molecules that actually translate the language of RNA into the language of proteins. Of the 64 possible mRNA codons, or triplet combinations of A, U, G, and C, 3 specify the termination of protein synthesis, and 61 specify the addition of amino acids to the polypeptide chain. Of these 61, one codon, AUG, also encodes the initiation of translation. Each tRNA anticodon can base pair with one or more of the mRNA codons for its amino acid. For instance, if the sequence CUA occurred on an mRNA template in the proper reading frame, it would bind a leucine tRNA expressing the complementary sequence, GAU. The ability of some tRNAs to match more than one codon is what gives the genetic code its blocky structure. As the adapter molecules of translation, it is surprising that tRNAs can fit so much specificity into such a small package. Consider that tRNAs need to interact with 3 factors. 1, they must be recognized by the correct aminosyl synthetase, see below. 2, they must be recognized by ribosomes. And 3, they must bind to the correct sequence in mRNA. Ribosome structure is shown with the E site, P site, and a site. Though a site is labeled aminosyl, the P site is labeled peptidyl and the E site is labeled exit. As ribosomes move along mRNA, tRNAs with amino acids attached bind to the sites. The polypeptide is assembled at the P site, and the empty tRNA exits from the E site. Figure 15. 16. The ribosome and its function. The ribosome is responsible for translating the mRNA into protein. The ribosome consists of a large and small ribosomal subunit. Assembly of the subunits on the mRNA forms 3 tRNA binding sites. B. During translation, charged tRNAs enter the acceptor site, and the anticodon on the tRNA base pairs with the codon in the mRNA. After the incoming amino acid forms a peptide bond with the growing polypeptide chain, the ribosome will move 3 nucleotides toward the 3 end of the mRNA. This movement will transfer the tRNA with the growing polypeptide to the peptidyl tRNA binding site and allow the empty tRNA to exit at the exit site. Credit: Rao, A., Ryan, K., and Fletcher, S. Department of Biology, Texas A&M University. Aminosyl tRNA synthetases. The process of pre-tRNA synthesis by RNA polymerase III only creates the RNA portion of the adapter molecule. The corresponding amino acid must be added later, once the tRNA is processed and exported to the cytoplasm. Through the process of tRNA charging, each tRNA molecule is linked to its correct amino acid by one of a group of enzymes called aminosyl tRNA synthetases. At least one type of aminosyl tRNA synthetase exists for each of the 20 amino acids. The exact number of aminosyl tRNA synthetases varies by species. These enzymes first bind and hydrolyze ATP to catalyze a high-energy bond between an amino acid and adenosine monophosphate, AMP. A pyrophosphate molecule is expelled in this reaction. The activated amino acid is then transferred to the tRNA, and AMP is released. The term charging is appropriate, since the high-energy bond that attaches an amino acid to its tRNA is later used to drive the formation of the peptide bond. Each tRNA is named for its amino acid. Amino acid and tRNA enter the active site. In this case, the amino acid cysteine, abbreviated size, and size tRNA bind to the active site of aminosyl tRNA synthetase. A TP is consumed, producing a MP and 2 inorganic phosphates. As a result, aminosyl tRN, a synthetase, catalyzes covalent bonding of cysteine to size tRNA. Figure 15. 17. Charging of tRNAs with correct amino acids. Aminosyl tRNA synthetases catalyze covalent bond formation between the tRNA and the correct amino acid in preparation for translation. Because there are multiple amino acids, there are multiple different tRNA synthetases. All of the synthetases require energy in the form of ATP to make sure the correct amino acid is attached to the tRNA with the correct anticodon sequence. Credit: Rao, A., Ryan, K., and Tag, A. Department of Biology, Texas A&M University. The mechanism of protein synthesis. As with mRNA synthesis, protein synthesis can be divided into 3 phases: initiation, elongation, and termination. The process of translation is similar in prokaryotes and eukaryotes. Here we'll explore how translation occurs in E. coli, a representative prokaryote, and specify any differences between prokaryotic and eukaryotic translation. Initiation of translation. Protein synthesis begins with the formation of an initiation complex. In E. coli, this complex involves the small 30S ribosome, the mRNA template, 3 initiation factors, IFs, IF1, IF2, and IF3, and a special initiator tRNA called tRNAfmet. In E. coli mRNA, a sequence upstream of the first AUG codon called the Shine-Dalgarno sequence, AGG, interacts with the rRNA molecules that compose the ribosome. This interaction anchors the 30S ribosomal subunit at the correct location on the mRNA template. Guanosine triphosphate, GTP, which is a purine nucleotide triphosphate, acts as an energy source during translation, both at the start of elongation and during the ribosome's translocation. Binding of the mRNA to the 30S ribosome also requires IF3. The initiator tRNA then interacts with the start codon AUG, or rarely GUG. This tRNA carries the amino acid methionine, which is formulated after its attachment to the tRNA. The formulation creates a faux peptide bond between the formal carboxyl group and the amino group of the methionine. Binding of the F-MET-T RN-AF-MET is mediated by the initiation factor, IF2. The F-MET begins every polypeptide chain synthesized by E. coli, but it is usually removed after translation is complete. When an in-frame AUG is encountered during translation elongation, a non-formulated methionine is inserted by a regular MET-T RNA MET. After the formation of the initiation complex, the 30S ribosomal subunit is joined by the 50S subunit to form the translation complex. In eukaryotes, a similar initiation complex forms, comprising mRNA, the 40S small ribosomal subunit, eukaryotic IFs, and nucleoside triphosphates, GTP and ATP. The methionine on the charged initiator tRNA, called MET-tRNA, is not formulated. However, MET-tRNA is distinct from other MET-tRNAs in that it can bind IFs. Instead of depositing at the Shine-Dalgarno sequence, the eukaryotic initiation complex recognizes the 7-methylguanosine cap at the 5' end of the mRNA. A cap-binding protein, CBP, and several other IFs assist the movement of the ribosome to the 5' cap. Once at the cap, the initiation complex tracks along the mRNA in the 5' to 3' direction, searching for the AUG start codon. Many eukaryotic mRNAs are translated from the first AUG, but this is not always the case. According to Kazak's rules, the nucleotides around the AUG indicate whether it is the correct start codon. Kazak's rules state that the following consensus sequence must appear around the AUG of vertebrate genes: 5-GCC RCC OG3. The R for purine indicates a site that can be either A or G, but cannot be C or U. Essentially, the closer the sequence is to this consensus, the higher the efficiency of translation. Once the appropriate AUG is identified, the other proteins and CBP dissociate, and the 60S subunit binds to the complex of MET-tRNA, mRNA, and the 40S subunit. This step completes the initiation of translation in eukaryotes. Translation, elongation, and termination. In prokaryotes and eukaryotes, the basics of elongation are the same, so we will review elongation from the perspective of E. coli. When the translation complex is formed, the tRNA binding region of the ribosome consists of 3 compartments. The A aminosyl site binds incoming charged aminosyl tRNAs. The P peptidyl site binds charged tRNAs carrying amino acids that have formed peptide bonds with the growing polypeptide chain, but have not yet dissociated from their corresponding tRNA. The E exit site releases dissociated tRNAs so that they can be recharged with free amino acids. The initiating methionine tRNA, however, occupies the P site at the beginning of the elongation phase of translation in both prokaryotes and eukaryotes. During translation elongation, the mRNA template provides tRNA binding specificity. As the ribosome moves along the mRNA, each mRNA codon comes into register, and specific binding with the corresponding charged tRNA anticodon is ensured. If mRNA were not present in the elongation complex, the ribosome would bind tRNAs nonspecifically and randomly. Elongation proceeds with charged tRNAs sequentially entering and leaving the ribosome as each new amino acid is added to the polypeptide chain. Movement of a tRNA from A to P to E site is induced by conformational changes that advance the ribosome by 3 bases in the 3 direction. The energy for each step along the ribosome is donated by elongation factors that hydrolyze GTP. GTP energy is required both for the binding of a new aminosyl tRNA to the site and for its translocation to the P site after formation of the peptide bond. Peptide bonds form between the amino group of the amino acid attached to the A site tRNA and the carboxyl group of the amino acid attached to the P site tRNA. The formation of each peptide bond is catalyzed by peptidyl transferase, an RNA-based enzyme that is integrated into the 50S ribosomal subunit. the energy for each peptide bond formation is derived from the high-energy bond linking each amino acid to its tRNA. After peptide bond formation, the A site tRNA that now holds the growing peptide chain moves to the P site, and the P site tRNA that is now empty moves to the E site and is expelled from the ribosome, figure 15. 18. Amazingly, the E. coli translation apparatus takes only 0.5 seconds to add each amino acid, meaning that a 200 amino acid protein can be translated in just 10 seconds. Visual connection. Illustration shows the steps of protein synthesis. First, the initiator tRNA recognizes the sequence AUG on an mRNA that is associated with the small ribosomal subunit. The large subunit then joins the complex. Next, a second tRNA is recruited at the site. A peptide bond is formed between the first amino acid, which is at the P site, and the second amino acid, which is at the site. The mRNA then shifts and the first tRNA is moved to the E site, where it dissociates from the ribosome. Another tRNA binds at the A site, and the process is repeated. Finally, entry of a release factor into the A site terminates translation, and the components dissociate at the stop codon. Figure 15. 18. Translation begins when an initiator tRNA anticodon recognizes a start codon on mRNA bound to a small ribosomal subunit. The large ribosomal subunit joins the small subunit, and a second tRNA is recruited. As the mRNA moves relative to the ribosome, successive tRNAs move through the ribosome, and the polypeptide chain is formed. Entry of a release factor into the A site terminates translation, and the components dissociate. Many antibiotics inhibit bacterial protein synthesis. For example, tetracycline blocks the site on the bacterial ribosome, and chloramphenicol blocks peptidyl transfer. What specific effect would you expect each of these antibiotics to have on protein synthesis? Tetracycline would directly affect tRNA binding to the ribosome, ribosome assembly, growth of the protein chain. Chloramphenicol would directly affect tRNA binding to the ribosome, ribosome assembly, growth of the protein chain. Termination of translation occurs when a nonsense codon, UAA, UAG, or UGA is encountered. Upon aligning with the A site, these nonsense codons are recognized by protein release factors that resemble tRNAs. The releasing factors in both prokaryotes and eukaryotes instruct peptidyl transferase to add a water molecule to the carboxyl end of the P site amino acid. This reaction forces the P site amino acid to detach from its tRNA, and the newly made protein is released. The small and large ribosomal subunits dissociate from the mRNA and from each other. They are recruited almost immediately into another translation initiation complex. After many ribosomes have completed translation, the mRNA is degraded so the nucleotides can be reused in another transcription reaction. The first section shows a ribosome reaching a stop codon on mRNA. The stop codon reaches the site of the ribosome, and a release factor interacts with the site as well. The second section shows the polypeptide being released. 2 GTP molecules go through hydrolysis, leading to the third section. In the third section, the ribosome subunits are shown separating, and the RNA is no longer connected to it. Figure 15. 19. Translation termination is active. Translation is terminated when a stop codon is in the A site of the ribosome. Since there are no tRNAs corresponding to the stop codons, the release factor protein enters and catalyzes the hydrolysis between the last amino acid and its tRNA. This hydrolysis releases the free carboxyl terminus, C-term, of the protein. Additional factors use the energy in GTP hydrolysis to disassemble the large and small ribosomal subunits and mRNA. Credit: Rao A. and Ryan, K. Department of Biology, Texas A&M University. Protein folding, modification, and targeting. During and after translation, individual amino acids may be chemically modified, signal sequences appended, and the new protein folded into a distinct three-dimensional structure as a result of intramolecular interactions. A signal sequence is a short sequence at the amino end of a protein that directs it to a specific cellular compartment. These sequences can be thought of as the protein's train ticket to its ultimate destination and are recognized by signal recognition proteins that act as conductors. For instance, a specific signal sequence terminus will direct a protein to the mitochondria or chloroplasts in plants. Once the protein reaches its cellular destination, the signal sequence is usually clipped off. Many proteins fold spontaneously, but some proteins require helper molecules, called chaperones, to prevent them from aggregating during the complicated process of folding. Even if a protein is properly specified by its corresponding mRNA, it could take on a completely dysfunctional shape if abnormal temperature or pH conditions prevent it from folding correctly. In the cytosol, the mRNA and ribosome complex have a signal peptide that bonds with a signal recognition particle. Once combined, they interact with a translocation complex and signal recognition particle receptor protein in the ER membrane. The protein is then removed from the ribosome complex and goes across the ER membrane into the ER lumen. Figure 15. 20 proteins are co-translationally targeted into the ER for secretion. Proteins that will be secreted from the cell will contain a signal sequence at the N-terminus. The signal will be recognized by SRP as soon as the amino acids emerge from the ribosome, and the ribosome will be targeted to the translocation channel in the ER membrane. The rest of the protein will go directly from the ribosome across the ER membrane and into the ER lumen. From the ER, proteins can be secreted from the cell via vesicle trafficking. Credit: Rao A. and Ryan K. Department of Biology, Texas A&M University.